classes it is very clear to us about the structure of DNA, right ma, about nucleotide structure, about diversity of DNA both in prokaryotic organisms and eukaryotic, then about salient features of DNA, double helical structure of DNA, Chargaff rules, the genetic, the search for genetic information. We are clear with all the experiments, and then we have also looked into what? RNA world. Central dogma of molecular biology is also clear to us. But then once again we will revise, because today's concept is what? Replication, transcription and translation, right. So, today once again we will recall this. Here we go with, first replication followed by transcription and translation. As we have discussed earlier, all the three processes occurs in prokaryotes and in eukaryotes. In prokaryotes, all the three processes occurs in the same compartment. What is that compartment? Single compartment. What is that? Where is the location of replication, transcription and translation? Right. Cytoplasm, right. And then if you see in eukaryotes, three processes occurs in different locations. Replication, of course, from cell cycle and cell division also we have learnt that DNA replication occurs during S phase of the interphase, fine, right. And then transcription and translation occurs in the cytoplasm of the eukaryotic cell, fine, right. Replication, transcription and translation. Replication occurs inside the nucleus, whereas transcription and post-transcriptional changes also occurs inside the nucleus. Post-transcriptional changes happens only in the eukaryotic cell. Post-transcriptional changes does not occur for prokaryotic cell. So, up till post-transcriptional events will happen where? Inside the nucleus only, fine, right, in a eukaryotic cell. Last process, that is translation, will occur inside the cytoplasm. Now, here you have to remember the enzymes as well, which is very important. Okay, DNA replication, a new DNA strand is getting replicated. Of course, there is a big enzymatic machinery we are going to learn in DNA replication, but the major enzyme is DNA dependent DNA polymerase. DNA dependent DNA polymerase. DNA dependent DNA polymerase is the major enzyme. It has highest synthesizing capacity, like it can polymerize around 2000 base pairs per second. Okay, it can polymerize around 2000 base pairs per second, fine. And then if you see transcription, that is a new RNA strand is being synthesized taking DNA as a template. Okay. Here what is the enzyme that is synthesizing RNA beta? DNA dependent, DNA dependent RNA polymerase. DNA dependent RNA polymerase will synthesize the RNA strand. Clear till here? Any doubt anywhere? And in few viruses where RNA is the genetic material, what happens? Reverse transcription happens for the central dogma to occur in those viral particles, where RNA is the genetic material. What happens beta? RNA gets turned to what? DNA. This process is what? Reverse transcription, or teminism also we will call it as. Okay. Reverse transcription. Is the voice audible now? Reverse transcription or teminism. Fine. It is reverse transcription or teminism. What is the enzyme that is required here for DNA strand to be synthesized from RNA? It is RNA dependent DNA polymerase. RNA dependent DNA polymerase. Fine, it is RNA dependent DNA polymerase. Clear? Any doubt anywhere, ma? Yeah. Now we will move on to the first foremost step, that is replication. Before getting into the replication, you should be clear on 3 models of replication. We have 3 models. What are they? 3 models of DNA replication. First is conservative model. Second, semi-conservative method of DNA replication. Third will be dispersive method. Here we go. What is conservative method of DNA replication? For example, if this is the parental strand, one is from 5 prime to 3 prime, other from 3 prime to 5 prime, right? Anti-parallel nature, anti-polarity, fine, right? So, if this is the parent strand, complete parent strand, by utilizing the complete parent strand, a double stranded daughter DNA is synthesized. That is conservative method, okay? By utilizing the complete parental strand, 2 daughter strands will be synthesized. This is conservative method. In semi-conservative method, what is the pattern is, if this is the parental strand of DNA, if this is the parental strand, fine, right? One parental strand will act as template, and to that template one daughter strand is synthesized. Synthesis will always be from 5 prime to 3 prime. Fine, right? And if another DNA, each DNA strand will act as a template here. Okay. For each DNA strand there will be a daughter strand. Fine. As such, 2 daughter strands will be synthesized. Here every strand is acting like a template strand. This is semi-conservative method. Now dispersive method of DNA replication. Here what happens is, if this is the double stranded DNA from 5 prime to 3 and 3 prime to 5 prime, every fragment of the DNA will act as, like, it acts as template, like bits and pieces of DNA will act as templates, and it will be like this. Bits and pieces of DNA will act as template, and daughter strands will be synthesized like this. Fine, right? This is dispersive method. Clear with conservative, semi-conservative and dispersive method of DNA replication. Of these 3 methods, the widely accepted method is semi-conservative method of DNA replication, which is proposed by Watson and Crick. Okay. Semi-conservative method of DNA replication is proposed by Watson and Crick. This is widely accepted. Who proposed double helical model of DNA? It is the same scientists again. Watson and Crick. Semi-conservative method of DNA replication is again proposed by Watson and Crick. Clear till here? Any doubt anywhere, ma? Yeah. Here we go with the experimental proof for semi-conservative method of DNA replication. Till here semi-conservative method is clear, right? Amongst 3 DNA replication models, semi-conservative method of DNA replication is widely accepted. Now here there is an experimental proof for semi-conservative method of DNA replication, which is given by Michelson and Stahl's experiment. Michelson and Stahl's experiment. And what is the model organism over here? Model organism is E. coli. E. coli is the model organism. Here you see my double stranded DNA. So what is this experiment all about? We heard about isotopes. What is isotope? Elements which differ in the atomic number, but the mass number will be the same. As such, the isotope, nitrogen isotope is taken. Here the heavy isotope is N15, normal nitrogen is having N14, right? So, we have two, that is N15 and N14. N15 is the radioactive heavy isotope, fine, right. So, when this E. coli is being kept into a high nitrogen bacteria, like high nitrogen nutrient is being provided to that, fine. That nutrient is having what? Heavy isotope of N15. So, that nitrogen is impregnated to the DNA of the E. coli cells. Okay. When the bacteria is placed into N15 medium, it is cultured in N15 medium, the DNA will have N15 isotope. So, here it is having N15 isotope, fine, right. Now, one generation time means one DNA strand replication time in E. coli is one generation time. One generation time in E. coli is 20 minutes, okay, 20 minutes. So, here N15 DNA is there, right. According to semi-conservative method of DNA, each strand should act as a template, right. So, when this heavy isotope is cultured for the next generation giving normal nitrogen medium, that is N14 for 20 minutes. When it is placed in the normal culture nitrogen medium, what happens is after 20 minutes you can see one heavy isotope and one light isotope, one heavy and one light. As such, hybrid is observed, N15, N14 hybrid nitrogen medium is observed. How come you can identify the hybrid? By centrifugation, okay. By centrifugation, taking cesium chloride as the centrifugation gradient. Centrifugation gradient, you can clearly see the dense nitrogen intermediate strains and the light nitrogen. Dense will be occupied at the base, right. So, N15 is observed at the base, okay, and light nitrogen sequences will be seen in the intermediate, which is N15, N14, fine, right. And light, complete light strains are seen at the top, that is N14, N14. As such, after first heavy isotope, when it is placed in the light medium, light nitrogen medium, you can see after first generation you can see two hybrids, where each of the strand is acting like a template strand. After second generation, after 40 minutes, means after 2 generations, you can see one heavy, 2 hybrid and one light chain. One heavy, one light chain and 2 hybrids. Okay. So, the questions from the application point of view, from this experimental proof, the questions will be asked. Okay. Good afternoon, please continue. N15 is the heavy isotope. Yeah. So, here what type of questions you will get, I will tell you. Application based questions. For example, if E. coli, which is having heavy isotope, that is N15, okay, when it is placed into N14 medium for 3 successive generations, the question can be asked like this, or 3 generations means 60 minutes. After 60 minutes, what will be the gradient? How many heavy isotopes, how many light isotopes, how many intermediate isotopes are found? Like that the question can be asked. So, here if you see, when it is placed in the N14 nitrogen medium, you can observe like in the first generation. N15, N14 are possible, yes or no? N15, N14, fine. Now, each of the strain will act as a template. In the second generation, each of the strain will act as a template strand. Each strand will act like that. So, you take, and again in the second generation also it is placed in N14 only, N14 medium for 3 successive generations. So, you can take it like this right now, beta, N15, N14, N15, N14. Only light chain is added to this. So, here N14, 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 N14. Fine, right? So here you can see two intermediate chains, and two light chains. Two intermediate chains and two light chains. Clear? Now third generation. Third generation, again each strand will act as a template right now beta. Okay, when each strand is template, again it is placed in N14, N14 medium. Now you tell me it has two heavy strands. Okay, means two hybrids are possible, yes or no? How many light chains are possible? 1, 2, 3, 4, 5, 6. 6 light chains are possible. And there is no possibility of a single heavy isotope. This will be after 60 minutes or 3 successive generations when N15 heavy isotope is cultured in N14 medium for 3 successive generations. And this will be the output at the end. Clear? Any doubt still here? You are clear with the experimental proof, right? Now, look at the DNA replication. Okay. First I will let you know about the enzymatic machinery. Here we see the enzymatic machinery of DNA replication. Enzymatic machinery for DNA replication. First it is the enzyme that is helicase. Helicase, which is energy required enzyme. This is ATP dependent enzyme. This is ATP dependent enzyme. Fine, right? Helicase enzyme, what is the activity of this? This will cleave the double stranded DNA. Okay, helicase enzyme will cleave the double stranded DNA first. So first, immediately after cleavage, what happens? Two single stranded DNAs will be present. These two single stranded DNA, not to get reactive, to make them stable. Okay, single stranded binding proteins will come into picture. Single stranded binding proteins. They will bind to the single stranded DNA to make it stable. Fine, right? Now, two single stranded DNAs are stable. But at the time of uncoiling, a tension is developed on the other end, yes or no? DNA is negatively coiled, but because of the uncoiling DNA, tension is created over the other end where supercoils are formed, okay, on the other end. These supercoils will be eliminated and DNA will be made negatively coiled again by the enzymes topoisomerases. In prokaryotes, the topoisomerases are known as DNA gyrases. Okay. Third enzyme is what? Topoisomerases. Topoisomerases are again ATP dependent enzymes. Fine, right? Topoisomerases. They will make the DNA stable. The coils, the supercoils which are formed on the other end will be released again. After this topoisomerases, what happens is, here RNA primer will come and bind to 3' hydroxyl group of the template strand. For example, listen here, to both the single strands, let us suppose this is the 3' OH group is present. Okay. At this point, RNA primer will be present over here. A small RNA primer is synthesized. Here at 5' and phosphate group which is there, this will be binded over here. This will start the synthesis of the new strand. So this RNA primer over the other end will act like what? Starter of the reaction. RNA primer is added over here by what enzyme? RNA primase enzyme. Okay. So the fourth enzyme is RNA primase enzyme. This RNA primase enzyme will add what? RNA primer. Fine, RNA primase will be adding up what? RNA primer. Now, this RNA primer should be removed. Now, the hero enzyme will come into picture, that is DNA dependent DNA polymerase, which will synthesize the DNA at faster rate, like 2000 base pairs per second. Okay. Now, that DNA polymerase, DNA dependent DNA polymerase will remove this RNA primer and it will start synthesizing the DNA strand. So, the fifth enzyme is DNA dependent DNA polymerase. Clear? Here we go. So, two template strands are present. Okay. Let us suppose here if you see 5 prime to 3 prime. The strand which is synthesized towards the replication fork. By the helicase activity, a replication fork is formed, right? So, the new strand which is synthesized towards the replication fork is known as leading strand. This is known as leading strand. And the strand which is synthesized away from the replication fork is known as lagging strand. But synthesis always takes place from 5 prime to 3 prime here. Here also it is from 5 prime to 3 prime. Okay, synthesis is always from 5 prime to 3 prime. Now, the template strand you see, here this is from 3 prime to 5 prime, here also it is from 3 prime to 5 prime. Okay, the strand which is synthesized towards the replication fork is leading strand, and the one which is synthesized away from the replication fork is lagging strand. The one which is synthesized away from the replication fork, what happens is, as it is hanging outside, okay, there is no stability here. To make it stable, small RNA fragments are synthesized first. Okay, these are RNA primers. They are known as what? Okazaki fragments. Okay, these Okazaki fragments are later binded up with DNA ligase enzyme. Okay, they are later binded up with what? DNA ligase enzyme. This DNA ligase enzyme will join up the RNA primers, and this will be a lagging strand again. Lagging strand is away from the replication fork, leading strand is towards the replication fork. Clear with the enzymatic machinery? Take down quickly. And then here we see, so the strand which is synthesized towards the replication fork, I said you it is the leading strand. This is the continuous strand, or the synthesis is known as continuous synthesis. The strand which is synthesized away from this is lagging strand, which is discontinuous synthesis. Continuous and discontinuous, clear, right? So, from this you remember that for DNA replication, the entire DNA strand will get replicated, okay. But for transcription process, for RNA strand to be synthesized, only transcriptionally active sites will be transcribed to RNA, okay. See, the complete DNA strand is replicated, but from this only those parts which are transcriptionally active, these parts will be transcribed to what? RNA. For example, if I have to take this small fragment over here, okay, look at it. First, from 3 prime end to 5 prime end, this is the template strand, okay. 5 prime to 3 prime, this is the new strand which is synthesized. This new strand which is synthesized is addressed as coding strand or non-template strand or sense strand. Fine. Whereas template strand is known as non-coding strand or anti-sense strand. Okay. Here promoter gene, structural gene, which is at the terminal ends fixing up the structural gene at the center. What happens here? You see here, promoter and terminator. Promoter is always towards the 5' end of the coding strand. Promoter is towards the 5' end of the coding strand and terminator is towards the 3' end of the coding strand. If I have to replace the template strand place with coding and coding with template, then the positions of promoter and terminator will also be shifted. Remember, this is a statement from your NCERT. Okay. So, promoter and terminator sequences are always aligned with respect to what? Coding strand. Coding strand 5' end will be what? Promoter region, 3' end will be what? Terminator region. Fine. When we are shifting out the coding strand with template strand, then the sequence or the positions of the promoter and terminator will be shifted on the either ends. Clear? And at the center the structural gene which is there, this will be transcribed. Fine, right? Now we will look into the transcription process in detail. Before that you have to know few things, okay, about cistron. Okay, cistron is the region from the DNA which is transcriptionally active, which is transcribing, and later this cistron will be synthesizing what? Protein chain. Okay, cistron is the active part of the DNA. Okay, transcriptionally active part of the DNA is addressed as cistron. Like that, in between a promoter and terminator, if it is eukaryotic mRNA after transcription, in between promoter and terminator there is only one structural gene which is one cistron. Okay. So, in eukaryotes what is the phenomena? Which is monocistronic condition. Okay. Whereas in the prokaryotic mRNA, here the sequence is what? Polycistronic condition. Polycistron. What does this mean? In between a promoter and terminator, at the center you have more than one structural genes. Each structural gene will be coding for a protein, right? So, here multiple protein expressions are observed, which is known as polycistronic expression. Fine. And here each structural gene at the mRNA before the translation process, what happens? Ribosome will come and bind over here, right? Okay. So, the mRNA with multiple ribosomes attached to it is known as polysome or polyribosome condition. This polysome or polyribosome we have observed in cell biology, yes or no? So, here multiple ribosomes, when it is attached with multiple ribosomes, mRNA strand with multiple ribosomes over it is polycistronic condition. Polysome or polyribosomic condition. Polyribosome. Fine. Clear till here? This is monocistronic, this is polycistronic. Okay. Multiple expression, multiple protein expression. This is observed in prokaryotes, and this is observed in eukaryotes. Fine. Any doubt anywhere? Yeah. Now, about transcription. Fine, right? Here you see. Transcription process is initiated with initiation, elongation and termination. Look carefully with respect to transcription. Here we go. First with initiation, elongation, termination. Fine. Initiation, what happens at the promoter region? Okay. Promoter region, we will say promoter region is at what prime end? 5' end of the coding strand. At the promoter region, the sigma factor will come and bind. Sigma factor along with RNA polymerase. RNA polymerase will have these subunits. RNA polymerase will have these subunits, alpha, alpha 1, you can say 2 alpha, beta, beta 1 and omega, total 5 subunits, right. To these 5 subunits, when sigma factor is added to this is addressed as a holoenzyme. Fine. To these 5 subunits, when sigma factor is added up, this is addressed as what? Holoenzyme. Sigma factor along with RNA polymerase is known as holoenzyme. This will come and bind to which region? Promoter region, okay. And here this will start getting a complementary sequence from the template strand. Fine. The what codons will be copied? Template strands codons will be copied and a complementary sequence is synthesized. Complementary governs the process of transcription here. Okay, what is the main principle of transcription? Complementarity. Okay, principle involved in the transcription process is complementarity. Complementarity governs the process of transcription. And RNA, newly synthesized RNA, it will be from what? 5 prime to 3 prime again. It will be complementary to which strand? Template strand. It will be same as that of coding strand. Only in the coding strand thymine will be there. At the place of thymine, it will be replaced with uracil in the coding strand. Thymine which is present in the DNA is replaced by uracil in the RNA strand. Clear? Fine. And now in the termination process. In termination what happens? The newly synthesized RNA strand will be released out. Okay. At the same time sigma factor will be released out. Okay. But RNA polymerase will be still in the bubble. Okay. A transcription bubble is formed after initiation. During initiation what is happening? RNA polymerase along with sigma factor will come and bind there. Okay. At the region whichever newly RNA strand is synthesizing, at this region a transcription bubble is formed. Okay. This transcription bubble is holding the RNA polymerase still. Even after releasing the RNA, it is still holding the RNA polymerase. This RNA polymerase is pulled down by rho factor. Okay. RNA polymerase is released out or pulled out by what factor? Rho factor. Clear till here? Everything is fine, right? Sigma factor, RNA polymerase, newly synthesized RNA strand, and termination is rho factor. By rho factor. Rho factor will pull out the RNA polymerase. Clear with initiation, elongation, termination, everything about holoenzyme transcription bubble as well. Any doubt anywhere? Yeah. Now, this process happens inside the cytoplasm of prokaryotic cell and inside the nucleus of the eukaryotic cell. In prokaryotic cell, the mRNA which is synthesized will not have any intron-exon sequences, but then eukaryotes will have intron-exon sequences. Okay. Here comes the concept of post-transcriptional events. Okay. Post-transcriptional events. What are the 3 post-transcriptional events? Where they happen? Post-transcriptional events occurs only in the eukaryotic cell. Fine. Inside the nucleus. What are those 3 post-transcriptional events? First one is 5 prime capping, then 3 prime tailing and splicing. Fine. First, at the 5 prime region, methyl guanosine triphosphate will come and this gets aligned with the 5 prime region of the mRNA. Okay. What is that getting added, ma? Methyl guanosine triphosphate. It is methyl guanosine triphosphate. By what enzyme? Guanylyl transferase is the enzyme. This is by the enzyme guanylyl transferase. Fine, right? Now, next is tailing process. 3' tailing, 5' capping and 3' tailing. At 3' region, polyadenylation occurs by the addition of multiple adenine groups over here. Around 100 to 300 adenine residues will be added over here. 100 to 300 adenine residues, okay, at the 3' end. Now, splicing. Here can you see intron exon sequences? These are intron and these are the exon sequences. Fine. Remember, introns are inactive sequences, exons are expressed sequences. Introns are inactive sequences, exons will be expressed sequences. Here we go, see, these intron sequences, they will be spliced by the enzyme spliceosomes. How spliceosomes are formed? Spliceosomes are formed like this. The intron sequences will be coiled up like this. These coiled up regions are called as spliceosomes. These spliceosomes are spliced from the main strand by SNRPs. Okay, SNRPs. What are they? SNRPs. They will cleave this spliceosomes. Those are intron sequences will be spliced by what? SNRPs. SNRPs are nothing but snRNA with some protein sequences. Why this 5' capping and 3' tailing is done? Why 5' capping and 3' tailing is done? This is done, first thing is to prevent the activity of exonucleases on the mRNA strands. Okay? The very first keynote is what? This prevents the activity of exonucleases. Prevents the activity of exonucleases. It prevents the activity of exonucleases. And here next is what? Splicing. Splicing is removal of intron sequences. Okay, intron sequences are inactive sequences. They will be removed out by what? SNRPs, sRNA and proteins. Clear? Any doubt till here? So all these post-transcriptional events, where are they happening? Inside the eukaryote. So the first synthesized RNA immediately after transcription, this RNA is known as hnRNA, that is heteronuclear RNA, which is having intron exon sequences. After post-transcriptional events, it is having what? 5' capping, 3' tailing and complete exon sequences at the end. 5' cap, 3' tail with only exon sequences. Fine. This is known as mature RNA or messenger RNA. This RNA will be omitted from the nucleus to the cytoplasm. Okay? This RNA will be omitted from the nucleus to the cytoplasm. See, can you see here the mRNA? This is pre-mRNA. Pre-mRNA. Mature mRNA will be spliced through the nuclear membrane into the cytoplasm. Clear? Now here the translation process will be operated. Before you can clearly know about the translation process, you should be clear with the transcription RNA, that is transfer RNA, that is tRNA, and you should be clear about the genetic code. Okay? We will do that first and then we will move on for the translation process. So first, if you see beta, genetic code here, DNA or RNA carries all the genetic information, right? it is expressed in the form of proteins. How many proteins are present? 20 proteins are multiple proteins, each proteins like they have the combination of 20 amino acids. 20 different amino acids will form up the proteins. The information about the number and sequence of these amino acids form protein is present in where? DNA. DNA holds up all the information that is building up into protein. This information is passed on to mRNA during the process of transcription. Thus, genetic code is the interrelationship between nucleotide sequences. Okay, genetic code is the interrelationship between what? Nucleotide sequences of DNA or mRNA and the amino acid sequence of the polypeptide chain. Okay. It is an mRNA sequence, contains coded information. For each amino acid, how many codons are present? 3 nucleotides or 3 codons. A single codon will have 3 nucleotides. That is 3 bases we can say. Fine. The proposition and deciphering of the genetic code was very challenging because there are 20 amino acids. So here it requires the involvement of scientists from several disciplines like physicists, organic chemists, biochemists and geneticists. George Gamow is the scientist who is a physicist, but still he has delivered the genetic code with 4 bases and they have a code for how many amino acids? 20 amino acids. 4 bases, 4 nitrogen bases, right? And they have to code for 20 amino acids. The code must be a combination of the bases. This is the rule. Okay? And he suggested that to code all the 20 amino acids, the code should be made up of how many nucleotides? 3 nucleotides, and it was a bold proposition at that point of time, because a permutation combination of 4 into 4 into 4, that will be 4 cube, which is having 64 codons, would be produced, which is more than a number of required amino acids, more than the number of required amino acids. What are the key discoveries in decoding the genetic code? It was 3 scientists, those are Marshall Nirenberg, Mathai and H.G. Corona. They have deciphered the genetic code. They gave us proper information. They have cleared up whether the genetic code proposed by George Gamow is true or not. This was deciphered. How Nirenberg has used poly-U mRNA chain for the synthesis of one amino acid, that is phenylalanine. If you can see the table of genetic code, U will code for what? Phenylalanine. Okay. So, poly-uracil mRNA chain, poly-U mRNA chain, here it is coding for what? Phenylalanine, which is the first codon that is discovered. Next is Corona synthesized RNA with repeating sequences. He has synthesized RNA with repeating sequences to decode the multiple codons here. Severo Okawo, he is the scientist, Severo Okawo, he discovered polynucleotide phosphorylase enzyme which helped to synthesize RNA in the template independent manner. They have synthesized RNA in what? Template independent manner. Clear till here? Any doubt anywhere, ma? Clear, right? It is George Gamow who has proposed, and it is deciphered by Nirenberg, Mathai and H.G. Corona. Here you can see the table. Okay. It is very simple and easy to remember this actually. Now here our mRNA will be our standard point, where thymine will not be there. In the place of thymine, uracil will be there over the mRNA strand. UCAG, fine, which is over the mRNA, and here also it is UCAG, fine. So the first base which is proposed here is taken from this vertical section, first letter, okay, and second letter is from this horizontal section, okay. Just see this one box, U is picked from here and then from here, okay, and third letter will be UCAG. As such, all boxes are filled. Now let us take one random box over here, okay. Cytosine is taken for the first letter, vertical section, second adenine, and then UCAG. Okay. Like that, if you see 64 codons, of this 64, you have only 61 sense codons. Okay. And 3 will be stop codons. They will not be coding for any amino acid. Those are stop codons. What are stop codons here? UAA, UAG and UGA. These are the stop codons. UAA, UAG and UGA. And then here can you see UCAG? Fine. UCAG. You have understood, right, how the third letter is filled. Yeah. Now you see how to remember the amino acid and their code. As such, if you want to fill the entire table, it will just take 2 minutes of time. First box you can see it is phenylalanine and leucine. First 2 bases are what? Phenylalanine. Followed by next 6 set is completely leucine. And then 3 will be isoleucine. AUG is methionine, that is known. Last box is completely valine. Okay. Now second column is easy to remember for us. See, serine, proline, threonine, alanine. Easy, right? 4 bases, 4 amino acids. So 4 codons coding for 1 amino acid. That is why it is called as degenerate codon. Okay. Here you see third box, 2 codons are coded for 1 amino acid. UAU and UAC for tyrosine. Next 2 will be stop codons, followed by histidine and glutamine, followed by aspargine and lysine, and then aspartic acid and glutamic acid in the last table. Last column you see, first it is cysteine, followed by stop codon and then tryptophan, followed by arginine, total 6 amino acid, which is interrupted by serine in between. Serine you can see here in the second table, and here you can see. Last one will be glycine. Okay. Clear? Glycine, which is optically inactive amino acid. Fine. Clear till here? Any doubt anywhere now? Now you look at the features of the genetic code that are being proposed. Okay. Genetic code, here it is a triplet code that we know. Every codon has how many bases? 3 bases. Each codon has 3 bases. For example, UUU for phenylalanine, AUG for methionine, which is a start codon. Next it is commaless. No codon is having a comma in between. Codons are read continuously. There is no punctuation in between. Okay. Degeneracy. One amino acid is having multiple codons. If you take leucine, it is having 6. Serine is having 6. Proline having 4. Like that, one amino acid is coded by multiple codons. Like that, 6 codons you can see for leucine, serine and arginine. Then you observe beta non-degenerate codons. Only one codon will code for one amino acid. For example, AUG, which is methionine, UGG for tryptophan. Universal. The codon in all the organisms, it will be the same. It will not be unique for human beings or any other organism. The codon is same for all the organisms. It is universal. For example, UUU codes for phenylalanine both in bacteria and in human beings. Then start codon. Okay, AUG will begin translation, and this codes for what? Methionine. Next, stop codons will end the protein synthesis. We have 3 stop codons. UAA is addressed as ochre, UAG as amber and UGA as opal. Okay, ochre, amber and opal. These are the points to be remembered carefully. Universal, non-ambiguous means one codon is specific for one amino acid. Degeneracy, one amino acid can be coded by many codons. Non-overlapping, the triplet doesn't have any overlap. Okay, there is no comma in between. Next, polarity. Okay, the genetic code is always read from which direction? 5' to 3' direction of the mRNA. Now tRNA. tRNA is also known as adapter RNA or soluble RNA. okay, adapter RNA or soluble RNA, fine, right. Here you see the adapter RNA or soluble RNA, it has two structures, 2-dimensional structure and 3-dimensional structure, tertiary or secondary structure you see. Tertiary structure, which is 3D structure, is inverted L-shaped model, okay. This is like inverted L shape. Fine. And this is clover leaf model, which is secondary structure 2D, which is clover leaf model. Fine. 3D is inverted L shape and this will be 2D, that is clover leaf model. In any model it might be, at 3' end there will be one amino acid, okay, that is CAA, the sequence is what? CCA, okay. At 3' end there is an amino acid. At 5' end, close to the 5' end, you have DHU loop, fine. Close to 3' end you have T-psi-U loop, okay. DHU loop is amino acyl synthetase loop, okay, and T-psi-U loop is ribosomal binding loop, okay. This is ribosomal binding loop and this will be amino acyl synthetase loop, okay. Opposite to this 3' and 5' end you have anticodon, okay. Anticodon, this will be copying the code from the mRNA, from mRNA sequence this will be copying the codons, clear. This is 3-dimensional structure, that is inverted L shape, here it is 2D, that is clover leaf model, clear till here, any doubt anywhere? Which is the active structure in this is the 3-dimensional structure, that is inverted L shape, which is coding for the anticodons, like anticodon is copied, is 2D. Who discovered tRNA? That is Francis Crick. Till now whatever I have taught is the complete thing about tRNA, but here look into the theory ones, okay. How much percent of tRNA is present in total cellular RNA? Total cellular RNA is a combination of mRNA, tRNA and rRNA. In that total cellular combination, tRNA is 15%. tRNA is 15% of total cellular RNA, and this is known earlier as what? Soluble RNA, and it is also known as adapter RNA. It performs two essential bridging roles. One is anticodon arm and the second is amino acid, that is acceptor arm. Okay, anticodon arm will bind to the codon on the mRNA during the process of translation. Amino acid acceptor arm, this carries a specific amino acid. This will carry a specific amino acid. Clear till here? Any doubt anywhere? Yeah, next. Secondary structure and tertiary structure. Look at this carefully. Secondary structure is proposed by Holly. Tertiary is by Kim and Clue. Okay. If you don't remember scientists also, no problem, but you remember what is secondary structure, what is tertiary structure, which is active and which is copying for the code. Okay. Here it has acceptor sequence which has CCA sequence, attaches amino acid over here at the time of activation of tRNA. Next is anticodon loop. This contains 3 bases that pairs with the codon on the mRNA. Next is DHU loop, recognized by amino acyl tRNA synthetase. T-psi-C loop, this holds the ribosome, which is ribosomal binding loop. Next, it has an extra variable arm which is found in some tRNAs, okay, which is close to the ribosomal binding loop. Clear? Acceptor loop, anticodon loop, DHU loop and T-psi-C loop. Next, composition and special features. 3 prime end will end with CCA, amino acid will attach over here. 5 prime end, this starts with what? Guanine. Okay. 60 plus types of tRNA are here. Each is specific for one amino acid. One tRNA holds for one amino acid only. Initiator tRNA is what? For methionine, that is having what codon? AUG. And there are no tRNAs for stop codons. Stop codons has no tRNA. Here we go, whatever we have discussed is there over here. Just go with the table. If you are having any doubt, let me know. Now translation, you are clear on the genetic code, you are clear on tRNA. Now translation process will be easy for you. Same like how you have initiation, elongation and termination for transcription process. Here also you will be having initiation, elongation and termination. But this the very first step is activation of the tRNA. Amino acid along with ATP. First amino acid should get activated. Active amino acid, when it gets binded to tRNA, tRNA will be charged. So the first step of translation is charging of tRNA. Okay. So here you see amino acid along with ATP in the presence of the enzyme amino acyl tRNA synthetase. This forms a complex called as amino acyl AMP enzyme complex, which is activated amino acid. This binds to the tRNA and makes it a charged tRNA. Okay. Amino acyl tRNA complex is formed. A charged tRNA is formed along with AMP and the enzyme. Fine, right? So here first step is what? Activation of amino acid. Amino acid AMP enzyme complex is formed. Next is charging of tRNA, which is also known as amino acylation of tRNA. Okay, that is the first step. Next what happens is, let us suppose this as an mRNA strand from 5 prime to 3 prime. Okay, there are some sequences here. So in this complete sequence, you will have untranslated regions at the 5 prime end and at the 3 prime end, because translation always starts from the initiator codon. Here where is the initiator codon? AUG is the initiator codon. At this region, clover leaf model if I have to take, tRNA will come and bind. Okay, and anticodon will be copied. Okay. Here it is T psi C loop, here it is D H U loop, 3 prime end where CCA amino acid sequence will be there, and this is the 5 prime end. Prior to the tRNA, ribosome will come and attach, that is smaller ribosomal subunit, larger ribosomal subunits. As we are discussing about eukaryotes, larger subunit and smaller subunit, right now. In the smaller subunit region, it has 18S rRNA. This will bind to the mRNA sequence, and here you have 5.8s rRNA and 28s rRNA and 5s rRNA. These are the protein subunits which are present in the ribosome. We know ribosome has RNA and proteins, right? So, here what subunit will come and attach over here? 18S will recognize from smaller subunit. Then larger ribosomal subunit will come. This ribosome will have 3 sites in it, A, P and E site. At A site the tRNA will come and land. At P site this will copy the codon, anticodon will copy the codon. By the time tRNA reaches to the E site, here an amino acid will be synthesized, okay, and this will get exited from the exit site, and then ribosome moves towards the next amino acid. One more tRNA will come, land at the P site, copy the codon, and from DHU loop another amino acid will be synthesized, and then ribosome will move to the next amino acid. This happens in the template independent fashion from 5 prime to 3 prime end, from 5 prime region to 3 prime region in template independent fashion. Clear, right? Clear till here? So, this is the complete elongation step. How a bond is formed between 2 amino acids. We know amino acid will have carboxyl end, amino group, R group and a hydrogen group, okay, and this is coded to next NH3, amino group, carboxyl group, R group and H. Fine. So, here carboxyl group of first amino acid is binded to amino group of the second amino acid. Fine. Carboxyl group of first amino acid is binded to amino group of second amino acid. This bond is by the enzyme peptidyl transferase, which is synthesized from 28S rRNA from the ribosome. Okay. What is the enzyme which is required for the peptide bond formation? Peptidyl transferase. Clear till here? Here you see the elongation process which is given in detail. After complete ribosome formation, mRNA tRNA complex is formed, which is an amino acyl acceptor site at the A site. It is established next to the P site. Here it will be clear to you, see A, P, E site. Fine. A new amino acyl tRNA reaches the A site and this forms what? Codon anticodon bond. Peptide bond is formed between carboxyl group of first amino acid with amino group of the next amino acid. And here what is the catalyst? Peptidyl transferase is the catalyst. Okay. This is found in 23S rRNA in bacteria, 28S rRNA in eukaryotes. Yeah. Translocation. So, here ribosome moves on mRNA from codon to codon. Amino acids are added one by one. These are translated into polypeptide sequences and they are dictated by DNA and they are represented by mRNA. A translational unit in mRNA, here the sequence is flanked by a start codon and a stop codon. mRNA may have untranslated regions which is from 5 prime end to 3 prime end. Last is the termination process. Termination process, of course, this involves what? Stop codons. Stop codons like UAA, UAG and UGA. This requires GTP dependent release factors. Okay. What are those release factors we have? Release factor 1, release factor 2 and release factor 3. Eukaryotic release factors in eukaryotes, release factor 1, 2 in prokaryotes. These factors will terminate the translation and they will release the complete polypeptide from the ribosome. This is chain termination. Fine. So, this is the complete translation process. Okay. So, with this we have dealt with the complete central dogma of molecular biology, that is replication, transcription and translation. In our next session we will be looking into gene expression, human genome project and DNA fingerprinting, right.